you want me to turn it on now or wait till
Well,
I
I was
go?
going to say turn it on now and make sure
There we go.
life is good.
Yeah, there are people Zooming, so. Okay, I think we'll get started. It's my pleasure to introduce today's seminar speaker, Professor Brian Freeman from the University of Illinois, Urbana-Champaign. Brian and I met over Zoom, actually, when I gave a seminar at University of Illinois during the COVID pandemic. And while we work in different areas, I found him really fun to talk to because he seems so curious about so many topics, which actually makes sense because chaperones are involved in virtually every aspect of cellular function. So you have to be curious about a lot of things, I think, to do good work in that area. Brian got his bachelor's in microbiology from University of Michigan, Ann Arbor, and his PhD in biochemistry and biophysics at Northwestern. So he is a real Midwesterner. But after a successful postdoctoral stint with Keith Yamamoto at UCSF, he started his own independent career at University of Illinois in 2002, where he's now a professor in the Department of Cell and Developmental Biology. He also has affiliate appointments in both the Cancer and Genome Biology Institutes, attesting to his breadth of curiosity and his institutional commitment. And he has some very interesting ideas about promoting DEI initiatives at his institutions. And so, again, the breadth of thinking about different things and incorporating service into his academic life is impressive. He's the recipient of several honors during his career, most recently being elected a Fellow of the American Association for the Advancement of Science, although the honors I was most envious of were the single-digit scores on multiple NIH grants, which I have never seen. So Brian's for myself, I mean, I've seen him for other people because I've served on study section, but Brian's longstanding interests are in the conserved mechanisms by which chaperones guide the dynamic protein environment in our cells, with an emphasis on central nuclear processes. And today, though, he's going to discuss recent progress on defining the cellular substrates of Hsp90s.
Awesome. Thank you so much, Catherine. It's been an absolute pleasure today. It was wonderful meeting Catherine by Zoom and really getting to know her in person afterwards. And I've had just a terrific day talking with people. I think, if anything, the meetings were all too short because, as she said, I do like to chat and I maybe talk a wee bit too much. With that being said, thank you for the lovely introduction, Catherine. What you're looking at, I will talk briefly about it today, and I put it up there because honestly, I love this movie. It's something that keeps me, thinking about how stuff works. And what you're looking at, I'm sure most of you realize this is budding yeast. This is mom down here. This is the daughter cell that forms. The nucleus upon budding is a closed nuclear environment. The green, we have a cherry marking the nuclear envelope and the outer envelope of mom and daughter. And the green dot is actually one site of the DNA that's labeled with a GFP-LAC fusion. And what really fascinates me about it, and one of the areas of that my group studies, and I will talk a little bit about today, is watch when it gets over to the daughter cell and then suddenly it sort of zips all over the nucleus. And that's when the genome's being reorganized. And we know so little about how that process works. And my work on chaperones got us into that area. But I would have to admit, if someone says the word molecular chaperone to me, I don't think anything about the nucleus whatsoever. That's just not where my mind drifts. My mind really goes over, that got bright in a hurry, into classic sort of chaperone biology, and that is really, I see Betty back there, just did beautiful work along with Bill Welch showing that Hsp90 binds the nascent chains as they come out of the ribosome. And it's not that they guide folding, but they provide an environment, a friendly environment for nascent chains to fold so that those nascent chains as they grow won't aggregate with other nascent chains. And that's a beautiful function for certainly Hsp90. And of course, if a protein can't fold to its, and we'll talk more about this, the seeming native state, 70 helps deliver that to degradation machinery such as the proteasome, where Aaron Checkanover has spent a lot of time showing that that's another nice chaperone role. And of course, even if a protein does fold to a seeming native state, there's still chaperone usage. Proteins typically don't work as a single subunit. They work in protein complexes. Many of the chaperones contribute here, including GROYL or TRK homologs. And then there's the area that my group likes to spend a lot of time on. And we started with supporting metastable proteins, where Hsp90 has a large role to help create an activatable state was the sort of beginning hypothesis. 90 doesn't do this by itself, but it does it with a whole slew of co-chaperones. But here we already have 4 wildly different roles for chaperones in the cell. And that sort of starts to beg the question of like, all right, well, how are chaperones doing folding, degradation, metastable proteins, complex assembly? And I think to get an idea of how is a chaperone functioning in a cell, I always go back to sort of an old school principle of in protein folding. As the nascent chain comes down its energy landscape, it often will encounter unproductive folding intermediates. And that is where, I think the majority of us, including myself, think about chaperones, that chaperone binding or activity gets us over these energy barriers and leads us to my favorite misnomer, the native protein. And I say misnomer because proteins don't have a singular native state. They always have multiple native states. And those multiple native states are usually indicative of the work a protein does inside a cell. And so we think of there being energy barriers, again, between those different native states when they're trying to accomplish work. And you might be thinking like, what? What exactly are you talking about? And my group, when I started my own independent group, I started with what I thought was the easiest one to sort of get a handle on. And that is, if we think about transcription factors or DNA binding proteins, we don't think of them as being non-native, whether they're free or bound to DNA. These are both native proteins, and yet there's a clear energy barrier between the free and bound state. If you take a purified transcription factor, bind it to DNA, this complex has a half-life of typically on the order of 2 hours. There's a clear energy barrier, and I say that because inside a live cell, that complex has a half-life of about a half a second. So there's clearly a difference between in vitro and in vivo. work through, in my group, demonstrated that a small molecular chaperone called P23, also happens to be a co-chaperone for 90, could accomplish the change in those states. And what you're looking at is DNA anisotropy experiment where a transcription factor called MCM1 is bound to the DNA. If we challenge this fluorescent labeled complex with a non-fluorescent DNA oligo, nothing happens to the anisotropy value, and that's because it's a very happily stable complex, not dissociating at all over an hour period. And yet if we challenge with P23, we quickly drop down to the anisotropy value of the naked DNA. And it drops down sort of on the kinetic values of what we see in live cells. And it turns out that P23 itself regulates about 70% of all yeast transcription factors, which is a very sort of chaperone-esque role inside a cell. And when I say that, chaperones are generally doing very broad activities inside a cell. All right, but what about Hsp90? Which is the topic of today. So 90 was originally discovered because it forms stable complexes with metastable proteins, typically signaling proteins, be it kinases or aposteroid receptors. A lot of beautiful work from Dave Toft, Bill Pratt, early work led to a model where Hsp90 will bind to clients in conjunction with recent work from Johannes Buchner's group, showing that 70 will deliver those clients along with various co-chaperones to 90. With ATP and additional co-chaperones, we will eventually carry out late folding steps to lead to an activatable state of those clients. And then when we have client activation, say hormone addition, it will destabilize this complex and it will return to its cycle. For 90 to do this, it doesn't work alone. I've shown a few co-chaperones. In budding yeast, there are over a dozen co-chaperones. In human cells, there's over 20. It's a very complex system. And what was more intriguing to me is that Hsp90 itself is a highly abundant protein. It's about 2% of your protein mass, way outnumbering the number of metastable proteins in your cell. So what are we doing with all that extra Hsp90? And we didn't really have a good grip for that. We knew that 90 had a large interactome, and so there are 1,400 out of 6,000 open reading frames in yeast that are linked to Hsp90. But that linkage wasn't what you would think for a chaperone. It wasn't a physical linkage. We didn't know that these were physically associating. Most of these interactions were based on just genetic interactions between the various open reading frames and 90 inhibition. And they came out of 3 primary papers, all really focusing on the genetic or chemical interactions between 90 and various open reading frames in budding yeast. Now, these genetic interactions have been useful for identifying what does 90 physically interact with. So my group has certainly taken a large advantage of trying to figure out what does 90 do in the cell? What does all that 90 do? What are its clients? One of the things that we focus on were telomere biology, where telomeres, linear ends of chromosomes in eukaryotes, have to be capped to protect them from degradation. 90 will actually attack this cap, much like P23 attacking transcription factors, and dissociate it. But 90 does additional things at the telomere. It actually helps deliver telomerase, the enzyme that will extend the end, and facilitates elongation of that DNA. So it's doing a fair number of things at that DNA end, and a lot of it being transient interactions. 90 will work with other things. It will work with chromatin remodelers. But here, the chaperones are doing different things. Before I mentioned P23 kicks things off DNA. Here, P23 doesn't kick it off, but actually greases this machine to slide the nucleosome along. And that 90 will then attack RISC, and this is from the RISC chromatin remodeler, at the DNA binding subunits to kick it off the DNA. So we were able to use the genetic interactions to establish what does 90 physically interact with. But those 2 quick little examples was 5 to 10 years of work. where you have 1,400 things to truck through. Did we really want to keep taking that approach? The answer was obviously no. That was just, it was non-tenable. How can we get at what 90 physically interacts with. Because if we looked of 90 interacting with the capping complex or the chromosome remodeling complexes, we wouldn't find it as stably associated. When it does what it does in a very transient way. So we needed a way to stabilize what 90 physically interacts with. And I was once again inspired by Betty Craig. So Betty had used a non-natural amino acid called BPA to look at Hsp90's interaction with its partners in live cells. And I thought, well, I like that crosslinker. Typically, I'm not a big fan of crosslinkers, especially with chaperones. I always worry about, like, once I break them open, I'll capture artificial interactions. But here, BPA, we're going to integrate it into the Hsp90 structure. We're going to activate it with UV light, and that will create our crosslinker, and we'll only have the crosslinker when we are activating it with the UV light. And so we're controlling when and where we're going to have a crosslinker. And we took advantage of the crystal structures, or atomic information from various groups, including Dave Agard and Lawrence Pearls, to pick where we're going to place the different moieties of BPA within the Hsp90 structure. 90, this is a dimer of 90. Here's one protomer, here's the other one, N-terminal domain, middle domain, and C domain. And we did try a lot of other spots, but they seemed to interfere with function of 90. These seemed benign, and they seemed like really great spots for us to utilize. And one of the things that drove where we picked, what locations we picked, was we knew they were close to sites of protein-protein interactions, be it co-chaperones or the really limited amount of client interactions of where 90 would bind to a client. The CDK4 kinase was one of the few that we had information on. So we thought we could then take advantage of this BPA screen, incorporate mass spec to answer the following 4 questions. We could identify, first off, what are all the proteins Hsp90 physically interacts, and build out a really good, solid physical interactome. We could answer a really seemingly simple question that had not been answered. How does Hsp90 interact with so many different clients? And that's why we actually were covering the 3 different surfaces. We didn't know what Hsp90 used. There was some good clues that the middle domain was going to be used, but we needed to study it in farther depth. And then also, I naively thought, we'll be able to use this BPA as a tag to identify where 90 interacts with the client. Because BPA is a non-natural amino acid, I thought that would just fall right out of the mass spec data, and you'll see later we have some success with it, but it certainly wasn't to the level I was hoping. And then finally, of course, we wanted to discover new pathways that were governed by Hsp90. By knowing what it interacts with, we could then figure out what it regulates. And so we started all this with a proof of principle screen with Matthias Meyer at Heidelberg University, to figure out exactly what were the samples that we wanted to test, how did we want to go about it, and then eventually we did a large-scale screen with Alex Campos at Sanford Burnham Presbyterian. And what we ended up deciding to do is take wild-type Hsp90. We do grow it in the presence of BPA, but this doesn't have any integration of BPA, and this would allow us to isolate those stable the metastable proteins that stably interact with Hsp90, and then ones that contain BPA, we would capture both the stable interactions and the transient interactors, and then of course, importantly, all these are His tagged. We also use a non-His tag to screen out what were sort of our background hits on our beads and were interactors. Through this approach, we were able to identify about 1,100 hits. Roughly half of those are dependent upon BPA. When I say dependent, we either have to have BPA there, or we at least enrich over wild type levels. And if you read around the circle, you'll see Hsp90 is really interacting. Anything that's going on inside a cell, we're hitting. And unfortunately, with my level of curiosity, I like to float all over the cell and try to address where is 90 working within the cell. One of the things I was really happy to see is that it was about split, half and half of clients in the cytoplasm and the nucleus. And I say that because it wasn't too long ago that chaperones weren't even believed to have a presence in the nucleus. All right. So first things first, how does 90 interact with 1,100 different proteins? The client interaction surfaces that were known are shown here when we started this work. Tau, glucocorticoid receptor ligand binding domain, and CDK4 kinase. The contact residues with Hsp90 on one of the protomers, primarily in the middle domain, a little bit for tau off in the end domain, and a few client interactions down in the C domain. We thought, well, what we chose should hit most of those surfaces, but the prevalence really seemed to be on the middle domain. And when we look and ask what residues are capturing what clients, all the surfaces seem to be used. There is a small bias for the middle domains where they have about 350 hits, but the end domain is 225 and the C domains are around 200. And notice, in this quasi-Venn diagram, there's a lot of sort of co-usage of the different surfaces. And I think this is how 90 achieves such a broad binding capacity with so many different clients. It's basically a combinatorial use of binding surfaces. We have clients that are dependent only on the end domain, middle domain, or C domain. We have clients that will use both the N and the middle and C, or even the N and the C. We have some clients that use all three domains. And then, of course, this is all within a single protomer. We have ones that will cross the protomers. And that is how you have one protein that is able to reach out, or at least in part, reach out to almost 20% of all the proteins inside a cell. All right, now then we move on to what I thought was going to be a simple, straightforward question. We have this non-natural amino acid. It's chemically cross-linked to its client. This is going to get us what is 90 recognizing on a client to make it a client. And you might think, oh, we already know what chaperones recognize. They recognize short hydrophobic peptides. Yes, for Hsp90. And that's the only chaperone we actually know the rules for. We had no idea how 90 was recognizing a client, and we thought we could get there with that. And I totally convinced my student that this was going to be easy and doable. These were our non-natural amino sort of tags coming out of 90 where we put the different BPAs. And when she started looking, nothing. And we had to search and go through so many different computational approaches until she hit upon Mirox. And John B. Coley is a grad student doing this, and she just dug in until she found a program that could at least recognize the cross-linked peptides. And that got us basically the needle in the haystack, because mass spec data, there are just hundreds of thousands of peptides, and finding the ones that represented cross-linked peptides was really difficult, but it didn't get us. We had a lot of false positives in that, a lot of sort of nonsense information out of that. She figured out we had to add 2 more criteria. And then we could finally, with confidence, identify BPA-containing cross-linked peptides. Sty1 is a classic co-chaperone for Hsp90, and by adding in these 2, we could read across the sequence in Sty1, cross to an X, that's BPA, and then more sequence density from Hsp90 itself. Sty was a really nice one to hit because we knew it's one of the best well-behaved co-chaperones in terms of forming stable interactions with 90. I mentioned that 90 kicks RISC chromosome remodeling complex off the DNA. It does so functionally through RISC3, and we capture RISC3. So we're gaining sort of confidence, like, okay, these hits are actually real. We also hit BNI1. BNI1 regulates actin dynamics. I'll get to that story in a little bit. It's one of the clues that help us solve how chromosomes are moved in interface cells. To really sort of drive it home for us, were we getting valid hits? If we look at where we placed in the cross-linkers, in Sty1 I mentioned as being a really good co-chaperone, when we dock it on with the known sort of atomic structural information, wherever the yellow is where we get a cross-link in the structure of the co-chaperone, and the red is where we have a BPA moiety. And so these 2 moieties under here cross-link to those 2. There wasn't any structural information for Sty at the tail, but we did capture the tail with a BPA there. And so all 3 hits made sense where the co-chaperones should interact. And we hit a lot of different co-chaperones and chaperones, really convincing us and hitting some chaperones we didn't really expect. We actually, CCT is the trick Greuel homolog in budding yeast. We didn't expect to hit them all. So 90 really does look like it's a central hub for chaperone interactions. And when we dock any of the ones where we had that information, we could always make good sense of that's why we captured them where we captured them. And so our confidence that this looks like this is working and capturing physiologically relevant things was making us happy. Using Janvi's sort of combination of Mirox and her additional criteria, we have 1,100 hits. We didn't get all the BPA peptides, but we did get out to 180, a little over 180. And that, I felt, was enough for us to start figuring out at least some of the rules for how Hsp90 might interact with either co-chaperones, chaperones, or even clients. 24 of those hits are coming from co-chaperones and chaperones. When we ran for sequence conservations or any commonality, there wasn't any. So it wasn't simple like Hsp90 was recognizing a short hydrophobic peptide. But along the way, we realized, like, oh, intrinsically disordered regions. If you're a chaperone or co-chaperone, you're not so likely to interact with 90 through your intrinsically disordered region. But if you're a client, you sure are. So there's a preponderance of client interactions at intrinsically disordered regions. And some of that is shown here, because I had to say the field of intrinsically disordered regions can be a little rough. It's still a developing field. If you run, there are many different software prediction programs for where IDRs are found. We were fairly dissatisfied by using one, so we typically run about 8. And this is where we got a mass spec hit for NOP9. And all of them are predicting an intrinsically disordered region. The nice part about NOP9 is we actually have crystal structural information. And sure enough, it's a disordered region. And usually what we see is 90 is right next to not just not within an intrinsically disordered region, but it's usually right next to a structural motif like an alpha helix or a beta sheet. And we see that for wherever we had structural information. That is usually what we see, except for PRI1, where that is truly just within an IDR itself. All right. So that was actually, to us, fairly huge, because it's telling us how 90 is now recognizing these clients. And for us, it was making a nice sort of contribution to the chaperone biology. 90 is typically thought of as a protein that interacts with seemingly native proteins. And the question always was, well, how's that happening? Well, if it's recognizing IDRs, that's a really nice substrate for chaperones to bind to. And also may explain why so many proteins contain intrinsically disordered regions. Prediction software usually runs about 30% of all proteins have sizable IDRs. So are these sort of co-evolving so 90 can regulate those native proteins and sort of feed back on each other. Moving past that, we wanted to really drive home our confidence in our hits meaningful. And right away, I was pretty pleased that about 2 years ago, we published a paper using DNA-seq to show that DNA footprints, when you inhibit 90, are radically modified. But we didn't have a physical interaction with, no one had shown physical interactions with all these transcription factors. Our network of transcription factors is rather large. Almost all of the hits in the transcription area are simply DNA binding proteins. And the work we had done before is within a DNA hypersensitivity site. If we look at the footprints in wild type cells, we see lots and lots of footprints. Some of them have consensus motifs. If we inactivate 90 for just 15 minutes, it's a radical change in those footprints. We use that information to look across the genome at all footprints, and at the 15-minute time point, we see typically a dramatic loss in the number of footprints, and that changes when we get out to 6 hours. It's a totally different sort of event inside the cell, and what we see is later on, we're basically just losing those transcription factors. And some of that came out in the biochemistry. If we look at steady state levels of transcription factors, late, there's a loss following inactivation of Hsp90. In this case, we're using a temperature-sensitive allele of Hsp90 called G170D, isolated by Sue Lindquist's lab. Or if we use a small molecule inhibitor called Ridicicol, at the 6-hour time point, there's a loss of any transcription factors, but at 15 minutes, there isn't any loss. And yet we lost DNA footprints. And so it was a bit of a quagmire, like, well, they're not being lost, yet they're not binding DNA. And it turns out 90 is required for a late folding step for many transcription factors. Within the transcription field, people have often just go with, well, I can't express my transcription factor in E. coli because the recombinant proteins are notoriously inactive. We went ahead and purified a few, RAP1, CBF, ABF, and MET31 in E. coli, and when it's just a transcription factor, there isn't any DNA binding activity, and that's because that transcription factor is never seen in a eukaryotic version of 90. If we simply expose it to yeast Hsp90, we gain DNA binding activity. And so we think that's the issue. 90 was required to physically fold these proteins, but if we look and ask, can we see 90 associated with any of these? We never could. And the BPA screen was really nice for that because it captured all these transient interactions and told us that 90 is actually folding all these proteins. Not all transcription factors require 90. Reb1 was a really nice negative control. All right. A couple years ago, we published a really nice paper, I think anyways, on chromosome motion. And we didn't mention in that work what really facilitated us to delineate the chromosome motion pathway was this BPA interactome. That was classically a really difficult and challenging area. People were trying to figure out how do chromosomes move within interphase cells. We had a fairly large contingent of players thought to be involved in chromosome motion. And chromosomes, of course, I'm sure most of you already know, have a really high level organization inside of cells. They are organized not only by chromosome territories, where each chromosome is placed in the same spot after every cell division, and they're also organized, in this case, this is nomenclature related to high C experiments of compartment A and B. You can think of compartment A and B as heterochromatin and euchromatin. Always well positioned after every cell division, going back to the same location. And the question became, well, how does that happen? How is such unique precision achieved? And one of the things that's, to bear in mind are chromosomes are huge and heavy polymers within the cell. So it's not a little task to position these long polymers. For us to address, does 90 have a role in chromosome reorganization, we took advantage of a system developed by Jason Brinkner at Northwestern University. He started this when he was a postdoc with Peter Walter. He's using the ENO1 gene locus that he can activate just simply starving cells for inositol. When you starve for inositol, you go from the center where you're inactive to the nuclear periphery, really associated with the nuclear pore where you are then active. And he's incorporated a LAC-I response element array where LAC-I fused to GFP will bind. This is where that movie I came up with in the beginning. And we can starve and move the LAC-I over to the nuclear periphery. And it always moves to the same nuclear pore, which is really fascinating. It's a really precise motion mechanism. The downside of this system is, yeast is a relatively small nucleus. This is a rather large spot. So the relative enrichment never looks great, but it's a very consistent twofold enrichment. It's enough to follow. And one of the nice parts of this system is if we impair different chaperones, here we're impairing P23, it's that chaperone that kicks transcription factors off the DNA, we don't get any movement. We saw the same thing with Hsp90, except now we're inhibiting with a small molecule inhibitor. We don't get any movement when the inhibitor is there, but if we take those live cells and simply wash it out, it moves. So 90 is actively contributing to the motion of that DNA locus. I'm not going to go too deep into this. This was a rather complicated story, and I'm going to just tell a couple of sort of points highlighting 90 and highlighting what we use the sort of interactome to dissect out how this pathway works. But the ENO1 locus has different transcription factors, ENO2 and 4, that will recruit in chromatin remodelers and will recruit in RNA polymerase. And then PUT3 is a transcription factor Jason had shown binds a very unique DNA element called the DNA zip code. And that is the element that tells this locus to go from the middle of the nucleus to that specific nuclear pore. We know ENO2-4 binds to these, and PUT3 binds to, well, ENO2-4 recruits in chromatin remodelers. This was information that was known before. ENO80 is going to have a really important role in motion. And then PUT3, we found, recruits in a myosin motor. And that myosin motor is one of the substrates for Hsp90, and so is ENO80. And both of those fell out of our interactome. Hsp90 is going to work with both P23 and another co-chaperone for Hsp4 to recruit in our nucleate actin at this site. And the myosin's then going to join the actin. It's going to be held there in part by the chromatin remodeler. And what 90 does to facilitate this pathway, if we do what's called an actin spin-down assay, where we're just adding this ENO80 interact with actin, and it does, it will spin down with actin into the pellet, because ENO80 contains actin-related protein subunits that were known to interact with actin, although the complex itself wasn't known before this. 90 really directs it towards whether it's interacting or not interacting with actin. And it changes its behavior quite a bit. Along with P23 will contribute to get a very different behavior pattern for ENO80 interacting with actin. And so that was one of the bits of this story and how we were able to show it. What I found really intriguing is it's not just a stable interaction. And we found that out by going to a single molecule approach where we were trying to recapitulate the chromosome motion with purified components. And so what you're looking at here, we did single molecule work with Paul Selvin's group. Actin is in purple, and in green are nucleosomes. In this movie, it's just the mixture of those two. And not surprisingly, you don't see nucleosomes interacting with actin. We didn't really expect to. It was really more of a negative control. If we add ENO80 with the labeled histone, there's a little bit more interaction, but not much. If we then add in 90 to that same one, now you see a lot of interaction of that labeled nucleosome bound to the remodeler, but it's really dynamic. It's constantly on and off, but it's always returning to the actin. This, we think, will become a very important sort of characteristic to the chromosome motion, and I'll talk about that in just a sec. So that's where 90's role with ENO80 is coming into play to favor interactions with actin. The other one is Myo3. We would have never really hit upon Myo3 if we hadn't already isolated it in our BPA interactome. Now, Myo3 is a myosin motor. It's to move cargo along actin. In this assay, this is called an actin gliding assay. If we've got our glass surface, we pin the myosin onto the glass surface, and then we put down fluorescently labeled actin. If the motor's moving, the actin will slide across that glass surface, which is why it's called an actin gliding assay. And we isolated myosin from yeast. We put it in the actin gliding assay, and we were severely disappointed. Nothing happened. So it seemed like we had destroyed our myosin motor, potentially, but if we put in 90 and Hsp4, the motor's perfectly fine. It's just always actively dependent upon 90 being there. And we think that is why we hit myosin. And the way this system works is we have short actin polymers, and I'm not showing that data to you today, but it's a highly dynamic polymerizing, depolymerizing actin network in the nucleus. It's short actin polymers. That transient interaction of Eno80 with the actin is important to move along that actin polymer as the myosin power stroke goes, pulling it over to that nuclear pore. What we're yet to identify is how that locus knows which pore to go to, and that's stuff that we're working on now. So those are things that either were published before we did the network, things that facilitated us publishing work, but that's already out. I wanted to tell you guys something new in terms of what 90 is doing. And the one that I'm going to focus on today is in translation. 70 has a big role with ribosomes, nascent chains. It was a known nascent chain binding protein, and the data is not out there because it was all negative data, but people looked hard. Does 90 have a role with nascent chains? And it doesn't. But it does have seemingly important role with the translating ribosome. And that's where I'd like to go. There was a few publications that said 90 had a role with the elongating ribosome. I never like to chase what somebody else has done, although I know there's a lot of important things in there for validation. I wanted to chase something we didn't know anything about. And a lot of our hits were for initiation factors. We didn't know anything about 90's role with initiating ribosome, so I was excited. The student got excited, and we asked, okay, what goes on when a ribosome initiates? Why might we get hits there? And a lot of things go on. There's a lot of sort of reasons for protein dynamics, protein RNA dynamics, but we have the 40S that's going to form primarily with what's called the multi-factor complex. And this is going to then interact with the RNA capping proteins to form an initial complex. And then this structure is going to move along the RNA until we get to an AUG. And the multi-factor complex is particularly important for finding that AUG. Once we get to the AUG, we'll have some more protein remodeling events. The 60S will join, we'll have the 80S, and off we go for elongation. All right, so there's a lot going on. What got us excited is that our BPA interactome had a lot of hits within these complexes, and I thought, okay, cool. This looks good. Let's move forward sort of in a cautious way and ask, well, is 90 associated with the 40S or the monosomes? And so we just did your sort of standard sucrose gradient. As Betty would have predicted, 70 is found both with all the 40, 60, the monosome and the polysome. Hsp90 is primarily, though, with the 40S, a little bit with 60 monosome, and then it teeters off pretty quickly. But it's there. And that gave us some, okay, let's keep going. Let's see, is there a role, an impact of 90 on finding the AUG? We took sort of a classic approach where we put in plasmids that are going to express ranello-luciferase with a canonical AUG or non-canonical start codon with firefly luciferase. So this will get barely used in wild-type cells. But when we inactivate 90, and we're going to use that G170T temperature-sensitive allele and shift to 37 degrees and inactivate 90 and just ask, how does the usage of that non-canonical go up or down? It goes up. So we start, the ribosome cares less and less about starting at a proper AUG. Great, all right, let's move forward into more high throughput ribosome profiling approach, and because there's a lot of other non-canonical start sites that could be used. So we joined forces with Natalia is a postdoc with Judith Freeman group, and we did the ribosome profiling. They did the bioinformatics. First time in my career, usually we do the bioinformatics. Ribosome profiling was a whole new nut for us. That was not so easy to crack, especially with the first question we wanted to ask. What is the ribosome profiling over the AUG? So if we do a meta-analysis just in wild-type cells, here's the 5' UTR. All the peaks are where the ribosome is pausing, and of course you have the highest occupancy over the AUG. And very nicely, when we do G170D, after 15 minutes, we lose that peak. And so when you inactivate 90, the ribosome really doesn't care so much anymore about finding an AUG. It's there on the RNA. It will lock onto the RNA, and it'll start making peptides. And we thought, well, who are the targets that are sort of contributing to this loss of finding the AUG? The multifactor complex is one that I had mentioned. This is associated with the 40S, and it's important for finding the AUG. Jami's going to use a sucrose cushion to look at monosomes and the 40 and 60S, what components are there. And in G170D, both without temperature rising or with the temperature rising, PRT1, which is EIF3B homolog in budding yeast, is always clipped. So it's not even dependent, and I have to say G170D is not the happiest mutant. That non-happy mutant already has PRT1 clipped, and if we inhibit in a different way with a small molecule inhibitor, we actually just see degradation of PRT1. And so we think that's one of the contributors to why it's no longer caring so much about finding a start site. But of course, there begs the question of like, well, is translation even happening when you inhibit 90? So we labeled nascent chains. And much to our surprise, 15 minutes at 37 degrees, where G170D gets inactivated, yeah, we're making nascent chains. If anything, we're making more nascent chains. We looked in our ribosome profiling data, and I'm going to take you a bit through this. This is the proper frame where you see in a heat map highest level of occupancy and wild type. These are read lengths. The ribosome does have different size footprints along that RNA that can tell you a little bit about what's going on, but in the coding region, in general, wild type is in frame as expected. And in G170D, after just 15 minutes, it's no longer in frame. That's across the entire coding region. It doesn't matter what footprint it is. And so that told us, oh, this is a significant problem that may actually correlate to some old clinical data. And it's not even terribly old clinical data. About 5 years ago, 5 to 10 years ago, there were 60 clinical trials targeting Hsp90. Those clinical trials all failed with the same sort of phenotype. All the patients exhibited a heat shock response. You can't inhibit a heat shock protein if you're making more heat shock protein, which explains why it failed, but it wasn't quite clear why it failed. At the time of that, there was one paper that suggested Hsp90 kept heat shock factor 1. It's the main transcription factor for delivering a heat shock response to cells in a quiescent state. And that heat stress would cause release, and HSF would go to the nucleus and up-activate genes. And so by inhibiting 90, they thought, well, maybe that's why we get a heat shock response. But the funny part is, no one really believed that paper. And I thought, well, is our data telling us why we would get a heat shock response? If we're making nonsense peptides, that essentially would mock a heat shock response. If that's true, then we should be able to sort of tweeze that out. And we took a shot at tweezing that out with the following experiment, where we use cyclohexamide in budding yeast as a way to inhibit translation. And we're doing heat shocks after 15, 30, or 60 minutes. These are controls. Heat shock, we're producing a lot of the yeast Hsp70 allele, SSA4. If we treat with cyclohexamide, the only thing we do is exacerbate that heat shock response. Not surprising. You want to produce Hsp70 to turn off the heat shock response. We're just keeping it on because we can't translate anything. The stress response can be brought on by more than just heat stress. You can add a proline analog, azide. And azide will get a heat shock response, but since it's a proline analog, when you add cyclohexamide, you don't get a heat shock response because you never incorporate that proline analog, so there's no sort of malfolded proteins to get you a heat shock response. If we do that same thing with ridicicol, we see the same thing. It's very translation dependent. If we inhibit translation, we don't get a heat shock response. And we now think we're in a good position to sort of go back and ask, oh, in those clinical trials that failed, is there a way we can increase the fidelity of of the ribosome for its AUG? And can we return to those clinical trials? All right. And I'd like to thank John Coley, is the grad student who did the majority of the work. She's carried out all the BPA, she's carried out the translation work. I didn't have a chance to talk about Audrey's work. She looks at the role of the nucleoskeleton and genome organization. Anna Mankiewicz is another student who looks at the role of 90 with tail anchor proteins, where we think 90 actually contributes to insertion of tail anchor proteins in the membrane. Nichus does aging studies. Anchi and Zlata did the former work on chromosome reorganization, or the transcription network. Former students that contributed to various areas of all of his work. Alex has been just a great collaborator in the mass spec work. Bill Breyer was our collaborator with Actin. Judith is with the Ribosome Profiling. Matthias helped us set up the right conditions for carrying out the BPA screen. Craig is a long-term collaborator on chromosome remodeling, and the Brinkners helped us immensely with one relocalization. And I need to, of course, thank you guys for your patience during my talk. Thank you kindly.
Thank you very much. Can you hear me? Yeah. So I'll open it up for questions. Does anyone have any questions? Okay.
So you implied that the effect on translation was due to direct interactions between Hsp90 and the initiation factors, rather than like an indirect effect with like the other 1,200 proteins that also interact with Hsp90. And I was wondering if is there a way to disentangle that issue, or even have you tried looking at initiation from an iris to see if there are changes for cap independent translation?
So we're now moving towards a more we're certainly moving towards an in vitro system to get at that, because it's always a concern for any of the chaperone work that we do. We typically try to take it all the way to a purified system. I know I won't be able to do that here, but at least take it to at least like a retic system and ask where we're not going to have so many sort of competing influences. One of the things that sort of brings a little bit of doubt, at least in my mind, that it's not some indirect effect, is we're inactivating after it's a 15-minute heat treatment. It takes 5 minutes to inactivate the 90, so whatever is happening is going to happen within 10 minutes. That certainly won't rule out kinases. It's still one of the possibilities. But that's, it's the best, I think, that we have right now. And I will feel better about it, and hopefully you will too, once we move to an in vitro system and ask, okay, can we see the same things?
Can the Auxin system work with Hsp90, or is it too concentrated to degrade
I think
it?
the Auxin system probably could work, but the small molecule inhibitors are good specific inhibitors. And we can use different 90 inhibitors. The one that we are using does have at least 2 other weak targets, but we can use C-terminal inhibitors and also sort of tweeze it out that way. But usually that's why we use both a temperature-sensitive allele of 90 and a small molecule inhibitors to attack it in 2 different ways and say, are we still seeing that same impact?
Thanks. So can I ask you to elaborate a little bit more on Hsp90 interacting with IDRs? IDRs are all the rage now, forming these liquid phase separation things. In the in vitro or any in vivo system, have people examined whether Hsp90 modulates the type of liquid phase separation that you see?
I don't know of anyone who's looked at 90 per se, because there wasn't any real reason to look before, but probably half a dozen of other chaperones have been looked at, and most of them do modulate that phase separation.
Do they promote more, or do they promote more LLPSs, or?
Usually it's less. It
okay.
dissolves them.
Dissolves them, okay.
That's, I would say, the general take on it.
Okay.
And a follow-up on the IDR question. So some IDRs are enriched in certain types, chemical flavors of residues, and I'm wondering if when you do your analysis, do you see any selectivity
of sequence. And that was the thing we focused on the most in the beginning, and nothing came out of it. Even if we sort of looked at it in different ways of just categorizing it in classes of amino acid side chains, and there's no sequence specificity to it. The only thing that we found was the presence of the IDR for the clients.
Okay. Then question about the I-80 and the interaction of the nucleosomes with the actin. That was it, right?
The single molecule stuff.
Yeah, single molecule stuff. So the I-80, I think it has some actin-related protein subunits in it, and I'm just wondering, like, do you know which subunits of I-80 complex are responsible for that?
All the ARPs contribute sort of mildly to the overall affinity. That was a bit of a disappointing answer. You can't get rid of all the ARPs in E80, but you can get one at a time, and the overall affinity went down just a little bit, depending on which ARP we got rid of. But it's not particularly satisfying, but I suppose biologically it might make sense, especially given the way it interacts with actin in such a dynamic fashion. Having multiple sites that all contribute would make that sort
multivalent
of behavior
right. Okay. All right.
more readily occur.
Thanks, Peter. I think there was one question before you, Dave, so I'm going to...
Hi, great talk. My question was also about IDRs. So you said you have looked at the amino acid composition, and I am really curious whether there is any hydrophobicity versus hydrophilicity pattern that you could identify, especially because you are saying that many of the clients are binding DNA, and there may be burial of nonpolar surface from the IDRs, perhaps. So I thought that that could be a way, perhaps,
Well,
for
the
Hsp90
DNA binding subunits
act.
of the whole interactome are not a large chunk of that interactome. So the little transcription pie of the pie chart, that was almost all transcription factors that bind DNA. But that is maybe, it's not even 10% of the entire interactome. And so those IDRs aren't even going to be near the DVD typically either. A few of them are. But again, we were left with no sequence, sort of common signatures in terms of amino acid sequences where we found 90 binding.
Not even a broad character like hydrophobic versus hydrophilic.
No, and that's what I was, I guess, poorly conveying. We broke it down to that, not even specific amino acids, but let's just go with, basis, basic or negative, and nope, we got nowhere. And that was very disappointing in the beginning, and it wasn't. I don't remember. I'm trying to remember how we even tripped upon to look at IDRs. And I think I believe John V was looking at, okay, let me take a look at crystal structures. What do I see there? And that's when she was finding, like, oh wait, it's where 90 interacts, there isn't actually structural information to go with. And then we went back and asked, what about IDR, sort of structural prediction software?
I see. And so within that context, can you say whether the function of Hsp90 among all of those 1,100 substrates is mostly to help final stages of folding or maybe hiding binding surfaces or something else?
Oh, I think that's a mix of lots of different things. Just in the limited time that we've spent asking functionally what does 90 contribute to a client, we see all sorts of different things. For today, I just happen to focus on DNA binding events, and they're, depending on the client, 90 might kick it off the DNA, or 90 might put it on the DNA. For most of transcription factors, it's putting it on the DNA, and that's because its partner, P23, is kicking it off the DNA. But then for chromatin remodeling, 90 is kicking it off the DNA. So we don't have a rule for what will 90 do to a DNA binding protein, let alone, in a lot of ways, what 90 does to Eno80 and actin is very comparable to what it does with some transcription factors in DNA. Actin is just a polymer, and it's helping to modulate whether it is or isn't interacting with actin.
So it sounds like it helps more to facilitate or disrupt interactions rather than help folding. Would that be accurate?
Yeah, I think there are clients where it is certainly helping. We found that with the transcription factors. That's a very late folding step required for DNA binding. And I didn't show you today, but by limited proteolysis, we see conformational changes in those factors. So that we believe is a folding one. But for a lot of things, it's really helping to promote protein dynamics and protein nucleic acid dynamics.
So is Hsp90 ATP dependent or not?
Hsp90 has an ATPase. It's a very sad ATPase. It hydrolyzes ATP about once every 15 minutes. And most of that ATP in the hydrolysis of it is really to control the conformational sort of what conformation 90 is. So I showed you the dimer comes together and I have an open surface. This is nucleotide free, this is ATP, this is ADP. It doesn't look like the chemical energy of the hydrolysis goes to the client, but rather to the conformational change within 90.
And how about P23?
P23 has no ATPase activity. And so what it does to all of its clients is just through binding energy.
And so how do you see that being dynamic, I guess? How does it change states? And do you see any cold-sensitive mutants in these proteins that get stuck?
So P23 is the deletion of P23 is non-essential in budding yeast. It's essential in mouse, but the yeast are cold sensitive. P23 does not directly, when it interacts, so when it kicks something off the DNA, it doesn't interact with the DNA binding domain. It always interacts with an adjacent domain that is often known to allosterically alter the DNA binding domain. A good example of that was with steroid hormone receptors. The ligand binding domain of steroid receptors is known to be able to shut down the DNA binding domain. P23 requires a ligand binding domain to kick it off the DNA. In an odd sense, it does that not just with DNA binding proteins, but GCN5, which is a histone acetyltransferase, has an ATA2 interaction domain next to the HAT domain that is used to control HAT activity. That's where P23 regulates GCN5 through that domain. It's always some sort of allosteric interaction, never with the activity that we're directly following. It's always using a sort of regulatory domain that's used elsewhere in the biology of that client.
But then to recycle it, it's just presumably using thermal energy.
Oh, when it recycles, once P23 kicks it off, so we can see P23 kicking off a transcription factor off the DNA. We can't see it bound to the transcription factor once it's free in the DNA. We have one nice example, MCM1. When it kicks, and I showed the data for P23 kicking MCM1 off the DNA. If we remove P23, MCM1 will not rebind to the DNA. But if we give it 90 will convert that conformation into a DNA binding form, and now it can happily bind to the DNA. But 90 won't join it to the DNA bound complex. It's just changing its conformation and allowing it to now bind to the DNA. So that's the only one where we have this sort of a stable state of it's free, but not DNA binding competent. 90 gets it to the competent state. P23 will kick it back off the DNA and leave it in a non-DNA binding competent way.
Does Hsp90's ATPase change when it's associated with clients? Its rate change? And
It can go up or down, as frustrating as that is. So some clients will get the ATP, and it's the usual. ATPase will go up with a client, but there are certain clients that will make the ATPase go down.
But presumably, the ATP binding and ATPase activity of Hsp90 are essential, despite it being function given it.
There was an initial publication that said ATPase was essential. And Johannes Buchner's group a couple years ago published a follow-up paper that said the ATPase is not essential. ATP binding is essential, but not its ability to hydrolyze it. Those yeasts are very unhappy and slow growers, but their life still goes on even if it can't hydrolyze ATP.
So this is really interesting to me. I mean, so something as fundamental as whether or not this thing hydrolyzes ATP is still somewhat controversial as to what it does?
I think the people that published that it was required agreed with Buchner and said, yes, it's ATP binding is more important than hydrolysis, and that the yeast can live if they can't hydrolyze ATP. But it is an extreme slow grower.
Yeah, I mean,
So
might as well
life
be
isn't
dead at
happy
some
without
point, yeah.
that.
Did you have another question?
So I guess I'm just wondering, so instead of thinking about Hsp90 may have evolved to interact with a certain set of proteins, maybe it evolved to just not interact with another set of proteins. And so like, if you look at the things that don't cross-link, does that give you any insight?
We haven't done that to any extensive degree.
It's like the negative of the picture instead of the actual.
That's a great angle to sort of go at it with. And I think we sort of are playing with that idea with tail anchor proteins, which I didn't talk about today, because we think for tail anchor biology, GET345 is this complex that delivers tail anchor proteins to the translocase, GET1-2. 90 delivers has a bias set that it delivers. But if you get rid of GET3, it'll deliver those. If you get rid of 90, GET3 will deliver 90. So they can cross over, but they certainly have preferences. So I guess in some ways we're pursuing that. But I don't have an answer for you yet, because that's relatively, we're like halfway through that project, I guess.
Okay, well, let's thank Brian for a stimulating talk. Lots of questions, so that was great.
Yay.
Got a lot of questions.
That's good.
Yeah, really good.
I sent you that paper. I don't know if
Oh,
you saw
thank you.
your email.
No, I haven't been able to check.
Turned out to be Ursula Bond.
Okay.
Why I forgot Bond. Ursula Bond.
Ursula Bond. Well, thank you.
From Scotland.